Welcome to lecture. Sorry I was late today. Start out with a clear question that's not going to count. Which of the following is true of alpha helices and beta sheets? Both are parallel, both are right-handed, both are left-handed, both present side chains as solvent, both wrap around each other. The correct answer is, of course, that both are presenting their side chains to solvent, sticking out from the alpha helices on the outside and sticking up and down from beta-pleated sheets. Today, we'll talk about the changing of structures. And of course, changing of structures means changing of functions. Two basic methods. There's a third one we'll talk about, but these are the two most common methods that are used. The first one is protein phosphorylation, which happens through kinases and phosphatases. And the second one happens through GTPases or G proteins, GTP-binding proteins. I'll probably mostly use GTPase for this course. So in the case of the kinases, it's a protein kinase. Ase means, of course, it's an enzyme. What it does, it takes a phosphate and moves that phosphate from ATP and puts it onto a protein. And once it has put it onto that particular protein, that protein is now modified because the phosphate has a large negative charge on it, and whatever that protein happened to be interacting with, be parts of the same protein or a different protein, is now going to be different because of that large negative charge. Once the phosphate has been put on, that changes again the structure. Then that phosphate needs to be taken off at some point. How is that phosphate taken off? It's taken off through the activity of a protein phosphatase. These protein phosphatases, all they do is they take the phosphate back off. And usually, in most cases, this is taking the phosphate off, changing the structure, and then turning off the protein itself. There are literally hundreds of protein kinases in any given cell, fewer phosphatases, but clearly this phosphorylation is extremely important in terms of how things are regulated in the cell. GTPases, on the other hand,
and chopping up that GTP goes to GDP, so guanosine diphosphate. So you have 3 to start with, and then it goes to 2. And again, it's that same kind of thing. Phosphate, 2 negative charges, going to change interactions either in part of the protein or interactions with the other part of the protein. This is a little different because the way that these proteins then start to bind back to, as you reactivate the protein, is get them to bind to GTP. And to do that, you usually need a particular protein, an extra protein. Again, not unlike thinking about phosphatases, which would be taking off the phosphate. This is a guanosine nucleotide exchange factor. That's that GEF on the screen up there. GEF. So guanosine nucleotide exchange factor takes out the GDP, puts in the GTP. In some cases, but this is actually relatively rare, you also have a GTPase activating protein, or a GAP. Just a quick aside here, another name from molecular biology is alphabet soup, just because of all the ridiculous abbreviations that we have. So GAPs and GAFs and you name all of it. So the GTPase activating protein, the GAP protein, speeds up the hydrolysis reaction, but the GTP-binding protein itself is the enzyme. So it's not the specific enzyme now. So start talking a little bit more about kinases and phosphatases. It's just another way of looking at exactly the same thing. In most cases, when you have a protein kinase, we talked about this last time really briefly, nucleotide kinases put phosphates on nucleotides, protein kinases put phosphates onto proteins. A protein kinase, that phosphorylation step, usually, as far as we're concerned, particularly for cell biology, activates that particular protein. There are exceptions as well. But that process is a protein kinase, and of course it's an ACE, so that's the enzyme, it's a protein kinase that puts on the phosphate. It's taken off by a protein phosphatase. Actually, absolutely needs the protein phosphatase in this case. There's not spontaneous hydrolysis. It's not the actual protein that's modified as a GTP-binding protein that makes it different. There are literally hundreds of protein kinases and protein phosphatases in our genome in most of our cells most of the time. So probably the majority of regulation that's going on in terms of changing structures of proteins. As we'll see much later, it's probably the fastest way to get change in some kind of activity is by making this kind of covalent change. And then again, the phosphatases is going back in the opposite direction. Again, it's an ACE, it's an enzyme, which does the job. The GTPases, and again, I'll probably call these mostly GTPases. People call them GTP-binding protein, but I like to use GTPases that emphasize that they have that enzymatic activity. So GTPases, again, they'll bind to GTP. When they're bound to GTP, they have one structure. After hydrolysis, that last phosphate is removed. That becomes a GDP-bound form, and that is going to change the structure. We'll see the change in the structure in just a second here. Generally, here at the bottom, we start out active when you're bound to GTP. There's hydrolysis that happens, and again, that's by the G protein itself. Once it's bound to GDP, it's now in a different conformation, different structure, different function, usually inactive. This process can be speeded up by those gap proteins, but not necessarily. You'll have this hydrolysis that happens anyway. However, this removal of the GDP and replacement by the GTP, that is the process to really need a helper protein form. It's not an enzyme because it's not actually stimulated in a chemical reaction. But that's your gap, your guanosine nucleotide exchange factor that helps out with that. So what do these changes in structure really look like? This is a bit of a cartoon model of that. Up on the top on the right, you have the GTP-bound form of the protein. As soon as you lose that last phosphate, you see a change in the structure, particularly of an alpha helix, red in the middle at the top here. That change in the alpha helix structure, just due to that loss of phosphate, causes a huge change in some of the other parts of the protein because basically they're not stuck on anymore. You don't have all those weak interactions that are helping hold the two different parts, in this case two different domains of the protein together. As soon as those come apart, that causes a big change. In this particular case, it's binding to tRNA, that release of the tRNA that takes place. We'll talk a lot more about this when we talk about translation later on. But this is a very general aspect. Again, the GTPases, GTP goes to GDP, you have a structural change, that causes a difference. There's one more that I wanted to talk about in terms of modifications to structures of proteins, also very much used for signaling, and any of you who were in Dr. Singer's class at all any period of time last term, or taking with him now, these are his favorite proteins, so I've got them. So this is the ubiquitination pathway. Ubiquitin is a small protein. It's extremely similar in lots and lots of different organisms, which is why it's called ubiquitin, because it's ubiquitous and very, very similar. It's about a 76 amino acid protein, but basically seems to serve as a marker, and a marker for all kinds of different things. Yes?
When you mean it's similar in most organisms, do you mean like the shape?
I mean the shape, the sequence, and its function.
So the sequence can be slightly different, but we still call it the same protein?
Yes, so it's going to be ubiquitous as one of those protein families that we talked about before. So that's, but ubiquitin is, forget the exact numbers, but it's usually 90% identical in terms of its amino acid sequence, which means what in terms of its structure?
Same.
It's exactly the same. So last time, anything more than 30%? Both going to have the same structure? So it's 90%, it's going to have the same structure. If it's got the same structure, what's going to have? It's going to have the same function. Yes, good. Okay, so that's ubiquitin. But again, mostly as far as we're concerned, it's just sort of serving as a signaling molecule here. But it's important that it's a protein. We'll get back to that a little bit later on. So in order to have this marker, and it's a covalent marker, which means there's a bond that gets formed between ubiquitin and literally the C-terminus, the carboxy end of the ubiquitin protein, to first a ubiquitin activating enzyme called E1, again, more alphabet soup. It's ubiquitin activating enzyme, and the way that happens is through coupling. Before we talk about different enzymatic activities, you have hydrolysis of ATP, but that energy basically gets saved by making a bond, in this case to the E1 protein, and putting the ubiquitin on it. This ubiquitin, once it's been put onto E1, which is the activating enzyme, then gets put onto, amazingly, creatively named E2. And that is now also known as the ubiquitin conjugating enzyme. And the ubiquitin conjugating enzyme, we should tell you what it does, it puts ubiquitin onto other things. And so that's basically as far as the ubiquitin is, that happens all the time for all of these modification processes. You always have an E1, you always have an E2. But then you've got lots of different proteins that ubiquitin gets attached to. And how does the cell know which of these to attach to? That's the E3 protein, or the ubiquitin ligase. I hate this term because it says ligase. You would think actually it's got some enzymatic activity. It doesn't. It's actually the ubiquitin conjugating enzyme that does that. But basically, the E3 protein is the adapter. It says, hey, this is the protein which we want to put a ubiquitin molecule onto. And by putting the ubiquitin molecule onto it, basically forms a peptide bond, because we have up here at the top, again, I can't see my pointer, so I won't worry about it. But the carboxy terminus of the ubiquitin, COO, interacts with an NH2. That NH2, however, is not at the amino terminus of the protein that is getting modified. Where is it? Where else do you find NH2? What were the 4 amino acids that you should remember?
Basic.
Glutamate, aspartate, lysine, and arginine. Lysine. Lysine's got those NHs that are hanging out on the outside. So ubiquitins always get linked to lysines, and it's this peptide bond that forms with that lysine side chain on whatever the target protein is. A couple of things happen with this. You can either have a single ubiquitin which is added right here, or conveniently ubiquitin has a number of lysines on its protein sequence sticking out on the outside. So you can actually have multiple rounds of this that take place. And so you can have a ubiquitin that gets attached to your target protein, which is derived from your E3 saying what you should be binding to, but then that ubiquitin can have an ubiquitin added to that, which can have an ubiquitin added to that, added to that, added to that, etc. And so through that whole process, you can end up with a polyubiquitin, and this polyubiquitin signal here is basically the EB signal. Tells the rest of the cellular system to take that protein, move it to the proteasome, which is basically the cell's garbage disposal, at least for proteins. We'll break down that protein into smaller pieces. Because it's absolutely critical for the cell cycle, but also for a lot of other things, and just keeping track of proteins. Looking a little bit more detail in this process, this is a structure of one of these ubiquitin ligase complexes that's missing the E1 protein, but the E1 protein is going to put the ubiquitin onto the E2 protein, which is shown here in red. Then you've got your big scaffolding protein, which is part of the ubiquitin ligase. This is what's going to interact with whatever target protein it is that you're going to be putting that ubiquitin onto. Many of them known as cullins, and again, that's exactly what Dr. Singer works on in his lab, these cullin proteins. So here's the cartoon of looking at that. What happens if your target protein in brown in the middle at the top there binds to one of the parts of the E3 protein that brings it close enough to the E2 protein where you can then have transfer of the ubiquitin from E2 to your target protein, and through that whole process. So this is what it looks like in cartoon form on the lower right. The upper right is what this looks like when you do cryo-electron microscopy, which is a sort of a new tool for looking at protein structures. And we may have a chance to talk about that right towards the end of the class. It's a really powerful technique which gives you not completely atomic resolution structures, but gets you pretty close. And in theory, it's a lot easier to do than getting atomic resolution structures. Before we finish talking about proteins, I wanted to talk about spaghetti. No, this is what the book gets into great detail about, but one of the really important things in terms of how proteins function is in interactions with other proteins. And when we talk about kinases and GTP proteins right at the beginning, a lot of that's about modifying the structure, and modifying the structure of that protein, modifying whatever it's interacting with. And so if you phosphorylate a protein, then maybe it can bind to something else that it otherwise wouldn't bind to. Or if it's not phosphorylated, it'll stop binding to that particular protein. And so it's in looking at all of these different proteins that interact with each other, and again, towards the end of the course we'll talk about how you can figure these things out, but just looking at all these proteins and how they interact with each other allows us to think about trying to pull apart this big mass of spaghetti to try and understand what's going on here. But one of the things that you can generate these plots from is literally by looking at what proteins one particular protein interacts with. So we'll start out here, way down here at this end, on the bottom of these purple ones. If you look at this one particular protein, it turns out it interacts with 1, 2, 3, 4, 5 other proteins. And then, this protein only interacts with that protein. And so it's a way of describing that. And if you're interested in studying how any particular protein really works, and you want to think about something that's probably really important for how the cell's actually working, you might want to think about looking at proteins like that blue one up there at the top. You see it's interacting with a whole bunch of other different proteins. So that's probably going to be a really important kind of protein. Whereas studying this one really, really intensively might tell you something about this protein, but it's not going to tell you much more about how the rest of the cell is going. So it's a way of thinking about this. And there are quite straightforward mechanisms to pull apart how all these proteins are interacting with each other. It's a lot easier to simplify this, and simplifying it, of course, you've got to look at the Ubiquitin system because that's what Dr. Singer looks at. But it's also what our textbook looks at. Start down at the bottom. On the right, with the green, the scaffolding protein, the Cullen protein. What is it interacting with? In many cases, it's interacting with various different proteins. Here you see all these F-box proteins. Those are the proteins that hang out at this end, the left end of the Cullen protein, and then start to interact with all those target proteins. All the target proteins it's going to be interacting with. These Cullens interact with E2 proteins, but it turns out not very many different E2 proteins. The E2 protein just up there on the right, the CDC, excuse me, which number is that? Too many different CDCs. You think the alphabet's bad? Try adding all the numbers to it as well. CDC 34, no, you're not going to have to remember that. But these different adapter proteins, the F-box proteins and the skip protein, SKP1 down here, one thing to notice, these are the ones that are really heavily connected to all kinds of other things. And of course, not surprising, these are the adapters that help bring in those other proteins to the ubiquitin ligase complex in order to get them modified. But at the same time, there are also a number of these things that are interacting with each other that have nothing to do with the whole ubiquitin modification system as well. So yes, a protein-protein interaction and seeing that these interact with each other is really important and interesting, but actually figure out what they do, drag them to experiments and go back and look at some of these things. So, next clicker question, which I guess we're not going to answer in questions because I don't have my materials with me. Process of linking ubiquitin to a protein generally requires 3 enzymes, 0, 1, 2, 3, or more than 4. It's E1 and E2. E3 is not an enzyme. So, 2 enzymes. C is the correct answer. So that's it for at least an overview of proteins. All of chapter 3 in what, a lecture and a half. Couple of important things that I just wanted to remind you of here. You have polarity from a molecular biology point of view, going from N-terminus to C-terminus. The individual amino acids are just as polar as a 1,000 amino acid protein. You can put together secondary structures. These are your alpha helices and beta pleated sheets. Putting those together is protein folding, giving you your 3-dimensional structure. Of course, it's giving you function. Any particular part of a protein that has a specific structure and a specific function is called a domain. And we'll talk quite a bit about domains as we move on through the rest of the course. Many proteins, particularly some of the bigger ones, and in fact we just looked at this ubiquitin ligase here, are made up of multiple different proteins. They've all got an N-terminus and a C-terminus. Usually they're going to be being made from different genes. These are multiples. And for the vast majority, of particularly all the polymerases and things we'll be talking about later on, these are multimeric proteins. There are multiple different polypeptide chains, usually again made from different genes that come together. Protein families, nice review down with ubiquitin there. These are similar sequences. We can have similar structures, similar functions. Usually they evolve through gene duplication, either within an organism, which is going to give you paralogs, or through speciation, which gives you orthologs. And we talked about allosteric and structural change. So any questions about proteins before we move over to the dark side? Yeah.
You said that E3 isn't a ligase. So is it not a ligase because it's not an energy dependent process, or it's not technically joining these two molecules together, it's just serving as a scaffold?
So it's just serving as a scaffold, not technically have the enzymatic activity. It's E1 and E2
That are joining.
that are joining that. It's the E3 is really holding the substrate in order to have it be bound to. Okay, DNA. I didn't get my DNA model, but I'll do my best to have it on Friday. I'll definitely have it next week. So my prop, I need it to walk around campus. So, talk a little bit about the history of Nukleinsäure. How's that German? No? Okay, nucleic acid, which is how this was originally found. Some of the building blocks, adenine, thymine, cytosine, guanines. Again, we should always review. Chargaff's rule, which interestingly enough, Edward Chargaff never called his rule. Everybody else called it Chargaff's rule, and he didn't. And he actually really didn't like Watson and Crick. He thought they were nasty upstarts and really didn't have a clue. But it was his data, which they used, that was really important for their structure. Transformation, the DNA was the genetic material. Nobody believed that DNA was the genetic material for a very long time. And it was only really in the pretty transformative experiments, sorry, pun didn't mean that pun there, the transformation experiments in the 20s and literally in the 40s where people figured this out. And this is, my parents were born before this. This is really not that long ago. Hershey-Chase experiment, what most people say was the real defining, yes, DNA is the genetic material. I think it was kind of the nail in the coffin as far as the DNA being the genetic material. But it was a really elegant experiment. Of course, it has to do with viruses we have to talk about. And then, of course, Watson, Crick. Everyone talks about Watson and Crick. Few people talk about Franklin, and even fewer people talk about Wilkins, even though he also got the Nobel Prize. So it was really all four of these people who were most important in terms of coming up with the model for DNA. The people who actually had data were Rosalind Franklin and Wilkins. The really smart, Watson and Crick, these were modelers. They literally put models together. So again, like my kids working with Lego, basically same kind of thing. Actually, Lego was developed almost exactly the same time. So nucleic acid, Nukleinsäure. Friedrich Michner in Switzerland at the time was basically looking around at chemicals and seeing what he could find. Yeah?
Sorry to interrupt the flow. It's just, should we be intimately familiar with what Griffith did specifically in the experiment, what Avery did, and so on with all these people for the test?
If I talk about it in a particular slide, which I'm going to do in about 4 more, yes. Okay. So, no, stuff that I talk about in lecture, but if there's something briefly here, I would say no. I wouldn't try and remember that it was, Friedrich Michner in 1871 who did this, but the person who was credited with discovering nucleic acid, yeah, I think that's reasonable. So, and basically just purifying whatever he could find nucleic acid-wise, or I should say big molecule-wise, And so he was looking for any kind of cell that had a lot of this particular material in it. And as I mentioned before, he was in Switzerland. And at that point, they actually had salmon swimming up the Rhine. They're all dead now. But at that point, there were lots of salmon swimming up the Rhine. Turns out that sperm is basically nucleic acid. There's very, very, very little else in sperm, a few mitochondria. But that's about it. Eggs are a different story. They're way more complicated. But in order to get large amounts of DNA, salmon sperm was one of the things that was very heavily used. We still use it today in the lab. Over 100 years later, salmon sperm DNA is a really easy way to get a whole bunch of DNA. So that's where it came from. First started with white blood cells. Turns out I couldn't get enough of those. Salmon sperm was really easy. And what I was able to find, just looking at this nucleic acid, was that it was big in terms of size, had a lot of phosphorus, and was pretty acidic. And acidic because of the phosphorus. Remember, acidity is giving off those hydrogens to water. And so that's where the acidity came from. A few years later, people figured out that, looks like these chromosomes are probably something to do with genes. Chromosomes are really nice because you can see them under the microscope when they've actually condensed. And you can look at different chromosomes. Different chromosomes seem to be moving, seem to be segregating together with traits. I didn't talk about Mendel, but it was about this time that Mendel's work was rediscovered. It was done considerably earlier than that. So people started to think that chromosomes and genes were pretty similar to each other. They figured out ways to separate chromosomes from everything else that was in the cell. And there's a lot of nucleic acids in these chromosomes, but also a whole bunch of proteins. And if anything, by weight, there was more protein than there was nucleic acid in the chromosomes. So everyone thought, hey, proteins, where's that? Proteins are way more complicated. They've got 20 different amino acids, usually. We'll talk about some exceptions later. There's always exceptions in biology. But 20 different amino acids, as opposed to these really, 4 boring nucleotides, C's, A's, T's, and G's. That can't be the genetic material. Genetic material is going to be complicated. And so for a long time, pushed, in fact, by a great deal by Arthur Levine, was that, nucleotides, pretty boring. Proteins are where it's at. Proteins also seem to have enzymatic activity. You could actually get enzymatic activity from a purified protein. It would make a lot of sense that this would be the actual genetic material. Levine, interestingly enough, everyone gives him a really hard time because he was the one who thought it had nothing to do with genetic material. He was the person who showed that it was 2'-deoxyribose, which was the sugar in DNA, and was a really, really smart biochemist. And in fact, probably part of the reason that he thought, and everyone else at the time thought, that there's no way that the nucleotides could actually be the genetic material, is it turns out in everyone's favorite sources of nucleic acid, salmon, sperm, and white blood cells, they're about the same amounts of A's, T's, G's, and C's. So, and that's all 4 of them, not particularly one versus the other. So, yeah, since they had so many, or say, there are only 4 of them, very similar amounts of each, pretty much everybody thought that what DNA was doing in chromosomes was that it was much more of a scaffolding. It was holding the proteins together. It was the proteins that were important. And you just had CATG, 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 which was just serving as a place to hold all these proteins together. And it was only through Griffith and Avery's experiments where they came across the thought that actually this might be something that's slightly different. And that's, of course, because they started to look at important and interesting things, but particularly those that are really diverse. Back to day 1, we talked about bacteria, archaea, eukarya, and all animals being ridiculously closely related to each other. Turns out that it was the study of bacteria that was really critical in terms of making this leap into showing that DNA was really the genetic material. So the first of those was the pretty famous Griffith experiment, and this was the whole rough and smooth. kinds of colonies. Hopefully this is review for all of you. But the basic message was, if you took a bacteria that would normally kill a mouse, virulent bacteria, mixed it together with a bacteria that normally wouldn't kill a mouse, then the two of those together would actually kill the mouse. It's actually not terribly surprising because you've got, you know, the bad guys as well as the good guys. But if you kill off the bad guys, then the mouse is perfectly happy. But if you kill off the bad guys and mix them with the guys that aren't bad, then the mouse dies. So this was the transforming principle. And the transformation that happened here was you took these bacteria, the S bacteria, or sorry, the R bacteria, rough, I always get confused, rough and smooth. We'll see actually the picture of this a little bit later on. So the rough bacteria don't make the mouse sick. The smooth bacteria do make the mouse sick. You kill the smooth ones, they don't make the mouse sick. But if you kill the smooth ones and mix them together with rough ones, the rough ones actually become smooth, which is really important. So that's becoming smooth. That's the transformation of rough to smooth. Okay, rough to smooth, who cares? But it kills the mouse. And so that was the main thing. And then a number of people tried to follow up on this, but at the same, this is almost exactly the same time that we had Levine saying, oh, it can't be right. So as 29, he says, no, you've got 2' deoxyribose with tetranucleotide. And 28, Griffith says it's transformation. Basically, most people thought he was crazy and said, no, this is some weird thing the bacteria do, and we don't even care about it. Fortunately, there were a few people, including Avery and his group, Avery, McLeod, and McCarty, who said, actually, we think this is actually kind of interesting. We're going to try and follow up on this. And the main thing that Avery's group did was to basically, instead of just saying, we'll take a killed S strain and mix it together with an R strain, we'll take that killed S strain and separate it into as many different pieces as we can, and then see if we can get that transformation to take place. And so literally the transformation was actually like this. So when they talk about rough and smooth colonies, this is in fact from their paper. These are those smooth, actually I would call them slimy colonies, and those are the rough colonies. So you start out with this, you need to get something from here that turns this into that. And so they did lots and lots of purification. They were pretty classic biochemists. And they found something that they thought was a lot like Michener's nuclein soil, you know, big and very acidic, etc. But the other thing that they tried really hard to do was try and then kill off that activity. So they used proteases, which chopped up all proteins. They used RNases, which chopped up all RNAs, and those seemed to work just fine. Didn't do anything to the transforming principle. On the other hand, they put in DNase, it was completely gone. So they really were humming and hawing about this, and it was only after Watson and Crick that people said, actually, these guys really were right. It was just a case of following up on some of this data. The other problem with this is it turns out that this kind of experiment doesn't work with all bacteria. People who said it's a weird thing that happens with some bacteria are right. And that's because this particular kind of bacteria is what's called naturally competent. And natural competence means it can take up DNA from the environment and put it into its own genome. And that's what's shown here. Down at the bottom is what we now know much more about this process. Turns out it's a capsule protein. It's the slime that you can see in the slimy colonies over there. There's a particular gene, the capsule-producing gene, that is then released when you break open these cells. And since these particular bacteria are naturally competent, they can pick up DNA from the outside and put it into their own genome, and therefore start out being rough like this and end up smooth like that, which of course gives you your now smooth, capsulated, and now also pathogenic morphology. Yeah?
do naturally competent bacteria, do they have any means for distinguishing or selecting any type of DNA that they're going to incorporate, or they just incorporate any fragment that they come contact with?
Yeah, so the question is, I'm trying to repeat this, I'm trying to record it, so hopefully it's working today. Put it up really quickly. Are competent bacteria competent for any DNA, or is it just specific DNAs? Interestingly enough, it seems to be any DNA, and it's probably, they actually use the DNA in most cases for nutrition. It's food, and it's only in a few cases that it ends up getting integrated into the genome like this. And in that case, there has to be some kind of selection to keep it there. And in this case, your pathogenicity, et cetera, the people in the lab are growing it this way. But it turns out, if you grow these cells for a really long time, making capsules energetically hard, so you actually lose that gene. And that's in fact where these first our morphology came from. So if you don't actually need a capsule, you actually get rid of it. So it's an evolutionary aspect. But probably for most competent bacteria, it's not so much picking up a new gene, it's more about, nucleic acids, I can use those. We can make more of my own. So that's the idea. Speaking of nucleic acids, Edwin Chargaff, working at similar times, a little bit later now, the late 1940s. You remember what was happening in the early 1940s? Yeah, so it's not an easy time to be doing a lot of this work. Was looking at the nucleic acids that it found, again, Friedrich Michner, a number of other people, and basically trying to separate out those individual nucleotides as carefully as he possibly could, and most importantly, getting a quantification for it. And as I mentioned before, the relatively crude techniques that were around at the time, it looked like there's just about the same amount of A's, T's, C's, and G's. So really, really similar amounts of these nucleotides in all kinds of different organisms, from bacteria to the salmon sperm to the white blood cells, etc. And there's, again, no way that all these really different things can have so different proteins, but so similar nucleotides. And the real key to this was looking at some bacteria. And this in particular case, these were some of the gram-positive bacteria, high GC gram-positive bacteria, we call them now, because it turns out they have a lot more guanine and cytosine than they do adenine and thymine, relatively speaking. And so what Chargaff found was that some of these different organisms, and again, it was mostly these, what he called the tubercle bacillus. We now know, Mycobacterium tuberculosis, have very different amounts of G's and C's relative to A's and T's. But what he found is that G's and C's were extremely similar to each other, and A's and T's were extremely similar to each other. But he didn't say that they were identical to each other. And that was where he and Watson and Crick started to have these arguments with each other. Because Watson and Crick said it had to be identical, and Chargaff was, well, it's not quite, it's like 0.01 difference. And so there were a number of discussions about that as well. But these are some of the background data which were available there. Then, of course, Hershey and Chase. It should be Chase and Hershey, excuse me. Martha Chase, who was a technician in Al Hershey's lab at the time at Cold Spring Harbor. This was also, again, in the late 1940s, where a number of people, particularly Max Delbruck and some of his cronies or colleagues, lots of these people were refugees from Europe who then moved to the United States. Many of them physicists, interestingly enough, who decided, okay, we're going to try and study the simplest kind of biology we can find, and the simplest kind of biology they could find were viruses. And particularly viruses of bacteria, and the viruses that we're very interested in just had protein and nucleic acid. However, it was clear that these were very heritable. So it turns out that one virus stays the same virus over long periods of time. You can actually see this by looking at how they kill cells. Turns out they do a very similar kind of pattern. This is something that can be maintained over generation after generation. They also can only replicate their genome when it's on the inside of another cell. This is one of my favorite pictures here, color EM, when the genome is being released inside the cell. So it's inside a cell that you get those replications taking place. In particular, viruses they use the so-called T-even bacteriophage. T1 is odd. T2, T4, and T6 were the ones that they ended up using. Turns out that they're basically identical to each other. T2, 4, and 6. 1, 3, and 5 are totally different for random reasons. But they had known, because the electron microscope was also developed at almost exactly that time, that the actual virion, the outside parts that are the outside of the cell, and most importantly for their experiment, all of these T-even bacteriophages replicated by bursting open the cell. So I'm going to leave this here with a picture of Hershey and Chase. Chase and Hershey here. Chase was a technician at the time that did experiments, ended up being a professor of biology. Later on, did some really nice work thereafter. I think it's a nice example of how, even the technicians, quote unquote, get some of the credit for this. Cold Spring Harbor Lab.
Clock tower at Cold Spring Harbor doesn't have the normal 4 directions, north, south, east, and west, but it has the 4 nucleotides, C, A, T, and G.